Have you ever looked at a snail? Like really looked? I hadn't, up until a few weeks ago, after I spoke with Dr. Mike Hadfield at the University of Hawaii. Dr. Hadfield loves snails. He has devoted a lot of his life to saving them from extinction. And aside from worrying about snails chewing up plants in my garden, I had never given them much thought. So I decided to look. I looked at pictures and videos, and even outside in my own yard. What I discovered is that kahuli, which is the Hawaiian word for snail, possess a sort of ancient, quiet beauty. Their shells can be a deep chocolate brown or a dazzling multicolored swirl. Most of them move slowly, sluggishly, we might even say. We have found evidence of snails' ancestors from almost 500 million years ago, a tribute to their evolutionary success. In Hawaii, there were once more than 750 species of kahuli that couldn't be found anywhere else in the world. At least 60% of those snails have gone extinct. There are 44 Hawaiian snail species listed as endangered under the Endangered Species Act, 41 in just one genus, Yakutinella. There are many more unlisted species that face serious threats. For decades, the U.S. Fish and Wildlife Service has supported snail conservation and recovery work in Hawaii, especially in partnership with the Hawaii Department of Land and Natural Resources. Some of that work involves captive breeding of the Oahu tree snail, a species that has been extinct in the wild since 1991. In this podcast, I interviewed Dr. Hadfield about his lifelong work to protect snails. While the work has been slow, it hasn't been sluggish. It's been filled with remarkable passion, energy, and resilience.
I'm Professor Emeritus at the University of Hawaii. I joined the faculty here in 1968, and as emeritus, I was able to do as I had appointments both in research and education. So my association was, all departments evolved in the last 20 years, and it was the zoology department to the biology department, and then finally the School of Life Sciences. My training at the University of Washington and at Stanford, so UW was for bachelor's and master's degrees in zoology, and my PhD was in biological sciences at Stanford. And I did the bulk of my time at Stanford at their marine laboratory, the Hopkins Marine Station in Pacific Grove. So I was really a marine biologist by inclination and training, and my focus here has been on the biology of marine invertebrate larvae.
Oahu tree snails are beautiful, with shiny, colorful shells. These kahuli have been described as jewels of the forest, and have been depicted as being able to sing in Hawaiian folklore. They occupy an important place in native Hawaiian culture, and traditional poetry, hula, Hawaiian lei, and chants featured these long-lived kahuli.
However, after I'd lived in Hawaii, about 5 or 6 years, you move to Hawaii and you immediately become aware of this incredible endemic evolution that went on here in plants, the silver swords, a lot of different plants, wonderful gradations of the birds, the honey creepers, and all those beautiful red birds and yellow birds, and then the tree snails. And, I was pretty much an invertebrates man. And in those years, in the early 70s, the graduate students in my lab were heavily focused on sort of energetics, comparing very closely related species of snails, let's say, some of which make very large eggs and have development right through to little snails that crawl away when they hatch, to species that produce millions of eggs that are spawned into the free and into the sea. And I'm trying to understand the evolutionary factors that lead to selection for these very diverse life history patterns. So I became aware of the acatenelline snails. So they run, from say a centimeter to 2 centimeters long. They're brightly colored. They have wonderful patterns of banding, circular banding. They're browns and whites and various shades of ochre and brown, and even into yellows and reds, pure black. So they're very attractive. They're very large. And they were particularly well known. And for instance, the genus Acatenella on this island where I live on Oahu was about 42 species. So that's an enormous radiation. We know the oldest rocks on Oahu are about 5 million years old. And so we have no idea when they first got here. But at any rate, that's been quite a rate of evolution.
Because of their geographic isolation, the native species that evolved on the Hawaiian Islands often can't be found anywhere else in the world. But these species have also evolved with fewer threats from predators or competing species. As a result, when invasive species do arrive in Hawaii, they cause immense destruction to native plants and animals. In the case of the kahuli, this destruction was caused by predators.
The very populations we were studying were disappearing. I mean, really, our snails were going away, and we were finding them dead on the ground. And that was because of predation. Predators on the snails are multiple. So, when the Hawaiians arrived here 1,000 years ago, there were Polynesian rats on their canoes, and rats eat snails. The sailing ships brought 2 species of rats, the roof rat and the wharf rat, from European ports, and those became established. So then we had 3 predators, 3 rats crawling around, eating the snails. Then in the 1950s, somewhere in the late 1930s, a thing called the giant African snail was introduced to Hawaii. It had made its way across the Indian Ocean, throughout the Orient, up into Japan, and it got here. So the giant African snail are big plant-eating snails that, get quite large. And by the 1950s, it was a garden pest. People were really upset because it was eating their orchids and their bromeliads and so forth. It was never really a major agricultural pest.
To deal with the African tree snail, other non-native carnivorous tree snails were introduced as a biocontrol. One of these, the rosy wolf snail, moved up into the mountains and started attacking native snails.
So what we found in the mountains, often we'd have a nice, robust population of Hawaiian tree snails we'd be studying, and then suddenly it would crash. And what we would find is their beautiful shells. The rosy wolf snail doesn't eat the shells. It simply shoots a big proboscis inside the shell and pulls the snail out and swallows it. So you find these beautiful shells on the ground. Rats do crunch up the shells, so you can always tell who's preying when you get there.
There have since been other introduced predators, including Jackson's chameleons and the New Guinea flatworm. The Oahu tree snail was once abundant. Then it nearly disappeared.
There is no evidence that the Hawaiian tree snails had any predators during this evolutionary period when they radiated. No predators. And you can imagine that that's very believable given this crazy non-adaptive life history, right? Slow growth, late maturity, low fecundity, and so forth. And still they were incredibly abundant. If you read descriptions of the snails in the late 1880s or early 1900s, they talk about them hanging from the trees like clusters of grapes. I mean, you could just go and trees were full of them.
In 1981, the service listed the Oahu tree snail as endangered under the Endangered Species Act. 10 years later, Hadfield collected 11 of the last species of these snails in the wild and began an experimental program to breed them in a lab. While it took some trial and error, Hadfield successfully started a captive breeding program. He later built exclosures, or outdoor pens, to protect the snails outside.
So that led me to then start trying to see if we could bring them into captive propagation, right? Farm them literally in the lab. knowing that the snails have to feed on molds, bacteria, whatever's growing on the surface of leaves, it's black molds that we most notice. We were bringing in bunches of leaves. We were keeping the snails in terraria. So we started trying, and we finally isolated one of these molds and got it growing in petri dishes on potato dextrose agar, beautiful layers. And the agar, you could sort of scoop it out of the petri dishes, and the agar would stick to the wall of the terraria, and the snails ate it off. I mean, they loved it. So we had another substrate. So we developed, finally, I was able to scare up the money to buy some environmental chambers, the first one, right? And what that was a thing that looks exactly like your home refrigerator, except you could set day-night cycles of light inside, and you could set temperature cycles of light. So you could make it warmer in the day and the sun came up, and then in the evening the sun went away and it chilled down. We eventually got it going, so we bought those plastic fish tanks you can buy at the pet store. We cut the bottom out and glued in window screen, so that they wouldn't fill up with water. We figured a way to put sprinkler systems in them to spray them from the top, so we could then run them on a day-night cycle, day-night temperatures, and wetting them frequently enough to keep them active. We climbed into the mountains at least every other week and brought a great big bag of fresh leafy branches of ohia trees in. We kept those then in the refrigerator, and we could take them out and replace them. We were growing our mold, and we basically set up a captive rearing protocol that worked very, very well.
The first exclosure Hadfield and his colleagues built was primitive, but effective. It was an electric fence about 4 feet tall and 4,000 square feet, with barbed wire at the top, mostly to keep out the human types of animals. The scientists immediately observed that while the snails outside the exclosure disappeared, the snail population inside continued to thrive. That was a few decades ago now. Here, you can hear Hadfield talking about the exclosures currently being used by David Sisko, a wildlife biologist with Hawaii's Department of Land and Natural Resources.
In the field, like Dave's predator exclosures now they are so state of the art. You know, they have big solar panels. They have closed circuit TV, so they can sit down in the center of the valley in civilization and see what's happening up at those things. If something sets off an alarm on the top of the fence, they can find out why. They can monitor them. They're just incredible compared to the first one we built up in the Waianae Mountains. The snails inside them are incredible, and they're clever enough that Dave and his gang, they're doing multiple species in them, things that were sympatric to begin with, but they're doing both tree snails and another family of snails that's endemic to the Hawaiian Islands of ground snails. They've literally snatched those from extinction and have hundreds of them now in the lab.
Hadfield emphasized the important role that his students and partnerships have played in his work. Hawaii's Department of Land and Natural Resources took over the captive propagation program and has poured an immense amount of time and resources into kahuli conservation. Hadfield also mentioned many of his students and colleagues by name, including David Sisko, who now works for the state, and Stephen Miller, who works for the service. There are dozens of others, many of whom started off as students in Hadfield's lab at the University of Hawaii. Hadfield credits Sisko and Hawaii's Department of Land and Natural Resources with making incredible progress towards conservation. The service has provided funding towards snail conservation work, including $2.5 million in competitive state wildlife grants to the state of Hawaii. In addition to releasing the snails back into the wild, there will also be snails living at the Bishop Museum in Honolulu and the Honolulu Zoo. The ultimate goal, of course, is to be able to have a world in which these kahuli can be out in the wild and thriving. But with so many predators still on the loose, that's not a reality today. Reintroducing the snails into exclosures is an important first step. The Endangered Species Act has played a critical role in the recovery of the Oahu tree snail. With hard work and collaboration, we can continue to make progress towards sustainable native kahuli populations. This species has played such a critical role in Hawaiian culture and our environment. We don't want to imagine a future without it.
It's important because it conserves biodiversity. Number one, that sort of is a big statement, isn't it? So what? Because we got a world that was full of all this incredible biology, which is the result, we know, of millions and millions of years of evolution, and on these islands it's unique. It isn't duplicated anywhere else. So if you're going to learn from that, you have to keep it around. So there's all this stuff you can still do. There's so much that can still be learned. And I mean, learned about how life evolves in nature, how evolution works, how biology works, how diseases work. And you can't do it if it's gone. If it's extinct, it's gone, and extinction is forever. It really is forever. And so that's what all these efforts are trying to prevent, is just the loss of biodiversity, the loss of basically living information.
My name is Lev Levy, and I'm a public affairs officer with the U.S. Fish and Wildlife Service's Pacific Region. Thank you to Dr. Mike Hadfield with the University of Hawaii and Hawaii's Department of Land and Natural Resources. The podcast music was provided copyright-free by the musicians on Pixabay and includes songs 7 a.m., Mellifluous, and Lullaby for a Frantic World by Juan Sanchez, Renacita by Mirko Baroni, and Molokai Sunset by Mike Cole. For more information on snail conservation, you can visit the Hawaii Department of Land and Natural Resources Snail Extinction Prevention Program website and the Oahu Tree Snails species page on the U.S. Fish and Wildlife Service website.